Namaste everyone. I wholeheartedly welcome you all on the third day of our Translational Ayurveda online lecture series. Today our topic is Ayur informatics, a roadmap to research work, and we have a very experienced speaker today, Dr. Prashant Gupta. Dr. Prashant Gupta, he has completed his post graduation MD in Ayurveda from a very renowned institute, BHU, and currently sir is working as assistant professor in the department of Ayurveda, All India Institute of Ayurveda, Delhi. Sir has more than 50 papers and over 300 citations, and he has over 12 years of teaching and research experience. Sir has many papers on this particular topic in silico studies and several such topics. So once again I welcome you, Dr. Prashant, and now the mic to you.
Thank you very much, sir. Thank you very much, first of all, to the Professor Joshi, sir, and my dear colleague, Dr. Akhilesh ji, who was actually my colleague at Raipur also. So, at the outset, I am very, very thankful to All India Institute of Ayurveda, Goa, which is actually our brother institution at Goa. So, I think before starting my discussion, I would initiate things from scratch. Am I audible or optimum?
Yes, sir. It's clear.
Okay. Okay. Thank you very much, sir. So, this term Ayur informatics is a very holistic and umbrella term where an, an, an informatic approach for an Ayurveda, utilizing informatics in Ayurveda is termed as an Ayur informatics. As of now, the formal definition is this. We have written in some papers some inclusive definition, but for initial step, it is something utilizing information in the Ayurveda. So we initiated our journey way back in 2000. I initiated my journey way back in 2018 when I joined my DHR fellowship at Pune. So few of my mentors and few of my colleagues were very enthusiastic towards that informatics. We learned there the this network pharmacology, and then I went for an Indian National Science Academy fellowship at IIT Mandi for this particular Ayur informatics, and there onwards, in 2018 onwards and 19 onwards, I was bit inclined toward Ayur informatics, and fortunately then when I joined All India Institute of Ayurveda at New Delhi, our director, erstwhile director, Professor Tanuja Nesri madam, has given very, very kind support to us, and we developed a lab which is now termed as Ayur Informatics Laboratory at All India Institute of Ayurveda. It is first of its kind lab. I'm not decorating that lab, but as of now, we are doing a number of things there, and among them, I'm going to discuss few things with you, which may be helpful for you. So, and this is my Dr. Akhilesh has given my brief introduction. So coming on to the first point, that what is called in silico studies. Anything which is done in computer is called in silico. And in silico studies and performed by simulation. This word comes from silicon, where actually computer use silicon chip, and whatever is done in that silicon chip is called in silico. So these in silico simulations are frequently used for multiple purposes in conventional science. Among them, the best one is screening the drug candidates, that which compound, maybe synthetic, semi-synthetic, or natural, is or having a potential for becoming a drug. Later on in 90s and 2000, then we got in pharmacokinetics, dynamics, then predictive toxicology, then predictive ADR, then predictive PKPD, then predictive immunology. So multiple things has arrived, and among them few we are using. Wholesale simulations is a area where now we are working. I'll discuss why we are working and how it is very, very important for Ayurveda. So the term in silico is now clear, that anything which we do in computer, for sure this is not a confirmatory research. It is just an predictive research which can give you a clue that yes, possibly among these 10,000 phytoconstituents, these 2, 3, 4, 5 phytoconstituents are working or possibly may work. Why this all in last 4 years? When we got, when we means when the word stuck with COVID, majority of the wet labs were not functioning. Wet lab, by saying wet lab, that means where we are using pipette or something and something, and dry lab means where we are using computers. So over the time, what we learned that majority of the drugs 2000 onwards, or in fact precisely 2010 onwards, majority of the drugs are ideally natural compound, gradually to semi-synthetic, and then to the synthetic one. And 74 antihypertensive drugs in last 20-30 years, 48 are derived from the natural products. 7 out of 10 anti-migraines are from traditional medicine, and we all know the story of anti-malarial drug, that artesunate artemether, which got uh, a Nobel Prize also, is a TCM's herb, which was actually used for a unknown type of the fever in the sub or rural belts of the China, and then it gradually came into the conventional science, and nowadays it is a very, very effective anti-malarial drug. So this all, why this treasure, this herb, this basket of herb is important, or this knowledge is very important for conventional drug development. So for Ayurveda, uh, I think no other Ayush fellows are with us, sir. I think only Ayurveda fellow are with us, no?
Yes, yes, only Ayurveda.
Sir. So for Ayurveda, if we are willing for a computational work, what we actually need? We need four things as of now: uh, software, hardware, skilled human resources, and the fourth is dedicated institutions. Software means a software which can utilize our phytoconstituents, our approach of basic fundamental approaches. Hardware means a very good GPU, or at least entry level GPUs for doing some computational works. Third, a person who can understand informatics and Ayurveda. And the fourth, which actually gives all three things at a single place. Fortunately, we have this all four places at All India Institute of Ayurveda at New Delhi and All India Institute of Ayurveda Goa also. Goa has also starting a dedicated Ayur informatics laboratory. My slides are changing now.
Sir.
So in last 4 years, what actually happened, if we take an example of COVID-19, what we learned during this period, that uh, this high surge in silico drug discovery actually take place in 19 onwards, when researchers screened a large number of phytochemicals to obtain a potent moiety against the severe SARS-CoV-2. Due to effective role of traditional medicine in treatment, what we learned, that yes, there is a hydroxychloroquine, yes, there is X medicine, yes, South Korean traditional medicine is using something, African countries are using some herbs for preventing or treating that SARS-CoV-2, and Indian Ministry of Ayush was also very, very working very, very hard to crack down that COVID-19. And during that time, we actually repurposed our Ayush 64 Kabasur Kudinir for COVID-19 management. And how Ayush Kwath has contributed for prevention, prophylaxis, and treatment, we all know now, because at least millions of people has taken that Ayush Kwath. And eSanjeevani portal has, and during those days when I published that paper, it was 1.2 crore people who has given their actually feedback on that mm, that Google form. So this all how it happened, let me give you some glimpse. Initially, when hydroxychloroquine was proposed, SCQ was proposed that yes, it could work in ACE inhibition. It was also some dry, some wet lab work. Then we attempted, then the Indian researchers also attempted, and initial publication was molecular docking analysis of potential compounds from Indian medicinal soup. It is Kabasur Kudinir extract with IL-6. So this was, you can see that this molecular docking is a computational work, and it was published in Bioinformation, which is a very reputed journal, where actually they says yes, if we are using Kabasur Kudinir, we can prevent that cycloscone, this cytokine storm. The IL-6 inhibition is being done by this Kabasur Kudinir. It was a computational work. Then again, a paper in Frontiers in Pharmacology in 2021, it got published, and there also they says through virtual screening. Virtual screening itself is another term for computational drug designing or computational uses. There also they says yes, there could be a good inhibition activity against main proteases of SARS-CoV-2. There also we proposed that yes, Kabasur Kurnire in the last left you can see recommended by Ayush Ministry can possibly work for COVID-19. These two papers were computational. And on behalf of these two papers, what you can see, yes, then a paper came into existence, that is open label comparative randomized control trial. When we propose such type of things in our IEC, there was means I'm not saying very, but at least a good resistance was there with yes, do you have any sort of evidence by using such things directly into humans? So computer has helped us a lot. And then we got this CTRI, and I collected it from CTRI. Then again, a paper for IU64 network pharmacology and molecular docking. This is also a good journal, although the impact is 1 and 2, but this paper also gave a base for clinical trial. And then you can see IU64 as an adjuvant to standard care in mild to moderate COVID-19. What I'm trying to say, again, a paper, open label randomized control pilot study. What I'm trying to say that once you got a computational results, it can give a support or a platform for your clinical research. Otherwise, people may ask you where in your Shastra SARS-CoV-2 is given, where and I'm not saying these questions are valid, but the reply is very easy. Yes, if we are using Oculus hirsutus in SARS-CoV-2, computationally it is working. Let me see in animal and humans, because Coculus hirsutus or IU64 is already in human use. So uh, so the toxicity study is not required. This is very beautiful paper which we published uh, from Raipur. In tribal areas of Raipur, this Coculus hirsutus, that Patal Garuni, is being used for unknown type of high grade fevers. So we attempted against SARS-CoV-2, and we got a very good and encouraging result, and it got published in nearly 5 impact publication Journal of Biomolecular Structures and Dynamics. And first time our lab appeared on publication. After some time, what I was just searching that CTRI, and then I saw that a very reputed company has filed a clinical trial to evaluate the efficacy and safety of the tablet of the purified aqueous extract of Cocculus hirsutus in the mild to moderate COVID-19 disease. I'm not saying that they are correlated, but for sure, when you are producing an evidence, if it is not working for you, it would work for someone who is actually working progressively or prospectively. So computation has that capacity. Now coming on to a few other things, when you are doing clinics, usual questions came to you in OPD that yes, sir, is your hub or your formulation would not interact with or would interact with the ongoing modern medication or an homeopathic medicine or any natural medicine or any supplementation? We don't usually do not have such answers. By computation we can create some good answers. Maybe that has to be replicated in wet lab and then to the clinics, but at an initial instance we can give yes, there could be a herb drug interaction, or there is no herb drug interaction, possibly no herb drug interaction. So this is another field where we can work. As of now we are working on drug interaction, but herb drug interaction is a computationally possible, otherwise it is very big research and 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 need some more money. So this can be done by computation also. And one more important thing that uh, which is usually been asked to us, that whether your drug is safe, minerals are safe or not. So safety and toxicities can also be predicted by computation, and now some beautiful softwares has been approved from US agencies also, that yes, if you are doing toxicity prediction by these softwares, you need not to have animal toxicity studies. If it is there, it's good. If not, then okay, we can work with this predictive values also. So what I'm trying to say, that predicting the oral acute toxicity of organophosphates, this is, we are not using organophosphates, but yes, we can use QSAR model for predicting our own drugs which has minerals. So this is another one area where we can do some good computation work. Metallic compounds interactions, their solubilities, their permeabilities of the drugs, metal ions, how they reduce or increase the absorption of the drug, these can also be predicted with the help of computation. As of now, what we are doing, we are now doing docking, dynamics, CAG, likeliness, drug data mining, QSAR, molecular dynamics, toxicity, gene ontology, and now we are doing multiple things also. This slide is almost 6 months back I prepared. Now we are doing protein homology also. We are doing principal component analysis. So we are doing all these things. So among them we'll discuss a few things with you, although this session is not a hands-on session, but if you are seeing it on computer, you can parallelly open a screen where you can type Swiss ADME. So those who can do it, please do. And this is an ADME. ADME full form is absorption, distribution, metabolism, and excretion. When someone asks us this, your curcumin, how it is behaving in the body, how it been absorbed, how it is distributed, how will it pass liver or not, whether it has been metabolized in multiple areas or in hepatocytes, how it has to be excreted. So on the basis of computation we can predict uh, that how that ADME of curcumin or any other phytoconstituent acts. And this is very simple. You just go to the Swiss ADME, and any compound, whatever compound, maybe it is a quercetin, maybe it is gingivirin, whatever Vesacin, or you just have to take their smileys from, you just type on Google smileys of curcumin, you would find smileys, just copy them and just paste in this area and run. And as soon as you run, you would find another picture which is like this, where you can find what is the solubility. Okay, what is the class moderately soluble, what is the GI absorption of that phytoconstituent, whether that phytoconstituent can cross the blood brain barrier or not, what are the CYP system of that phytoconstituent, means how the liver and how it will pass through the liver, and what is the mechanism in the liver. The CYP systems are being given here. And then a very important thing which is called drug likeliness, whether this phytoconstituent can work like a drug or not. So we have 5 types of qualifications. One is Lipinski qualification, Ghosh, then Weber, then Egan, then Muges. And you can find, I have taken an example of curcumin, uh, and then you can see, yes, all of the rules are saying that this could be a drug candidate. And then the bioavailability score, and then the other things which I'm not going to discuss. But for sure, let's say let's say you are using that curcumin as an oil, then you must have to check log kp value, that whether your oil, if you are using that phytoconstituent in oil, whether it is going to be absorbed or not, or skin permeability of that phytoconstituent is there or not. So these are the things which you can use for your wet lab. So this is a very important thing which we usually do before doing further computational works. Next, coming on to another type of software, which is a toxicity prediction software, where we actually do just one thing. Same, we copy that this smileys, and then just putting this area or a pubchem name, just searching. Let's say you just put an aspirin here and then search, you would find a open box here where you can see whether this herb or this phytoconstant or this synthetic compound, you want to check for hepatotoxicity, you want to check for carcinogenicity, immunotoxicity, mutagenicity, cytotoxicity, AR toxicities, ER-LBD toxicities, PPAR systems, nuclear factor toxicities, stress response pathways. And you just do one thing, you just click all. Here you can find all button, you just click all, and then start tox prediction. You just click start tox prediction, and you would find this type of analysis within 1 to 2 minutes. And now then you can see, yes, hepatotoxicity, it's green, yes, there is no chance or less chance of hepatotoxicity. Carcinogenicity, yes, it's non-active for carcinogenicity. You can see all are non-active, means our curcumin is much safer. Usually if something is this is, if not in the range, then it would be color changing, means it could be red or purple or dark red. Just let's I was preparing for hands-on, but I'm not sure how many of you have the access for good internet and a laptop. So just type protox on Google, then go for this page would open toxicity prediction, then this would open. Just copy canonical smile of your choice phytoconstant, then just type this would open, and just click on all and start prediction. The prediction would give you this picture, and this picture says yes, the LD50 value is nearly this. Whenever you are proposing your research in IC IRB, usually people ask you what is the LD value. I will not allow you to do your Anubhut Yog for directly clinical use. Then you can give this data for support. And toxicity class is given, yes, as 6 is very safe, 5 is quite safe, 4 is safe, 3 is maybe vulnerable, 1 is yes, possibly vulnerable, and 1 is for sure the toxicity could come. This software usually also gives the reproducibility and what is the prediction level, that whatever they are predicting, how much possible that it would work in the wet lab. So prediction accuracy is 100%. Sometimes if the structure is complex, then this prediction value is less. Now coming on to the main topic, which is usually now means bread and butter for computational studies, we saw computational molecular docking. Molecular docking is as simple as lock and key method. For us, although this may not work because we use a polypharmacology where multiple phytoconstituents act simultaneously on a single target or a multiple target. So multiple target, multiple formulation, the softwares are now not available. Maybe if we require or if we have good consumers, one day these software developers can give you such type of softwares also. As of now, we have this protein, we have X compound, and we dock. As in this case, we this is in phytoconstituent, this is in protein structure, and we are docking it. How we got to know that dock has to be done here? Because thanks to our conventional science researchers, they have developed such a good extent of computation or this protein studies that yes, we know now, we know that where the aspirin would attach in a, a particular type of protein, and then the cascading would come up and the result would be the blood thinning. So at that same particular area, if my phytoconstituent is also working and the doc score comparing to X synthetic compound or a known drug with my phytoconstituent is comparable, then it suggests that this phytoconstituent or a bioactive can work as similar as this synthetic compound. Uh, Although this is a slightly advanced slide, but as I know that nearly 500 people are looking this PPT, so few of them may be the senior professors also. For them, I'm just giving glimpse that how we calculate or how we reach towards protein and a key. Lock and key, we require lock and key. So just to get an key, it's very simple in conventional science that they have a, let's say they have a acetaminophen, they have a ivermectin, they have a hydroxychloroquine. They know there's the structure of that synthetic compound. But in our case, let's say we have a trifla. Trifla has 3 herbs and then at least 3000 phytoconstituents. How we will catch these valuable phytoconstituents? So we have two methods. We'll come on to key part first. How we will come on to the phytoconstituents. So one is just typing, let's say Terminalia tubula, and just typing it in the database, and you would find n number of phytoconstituents. You can catch them from there, and again to the next hub, to the third hub, and then compile these all phytoconstituents, and then doing the ADME whether this can cross the GI or not. And if let's say you are giving trifla orally and some phytoconstituents are not GI absorbable, then you can exclude them for the study, for further study. One approach is this, that you are taking the help of the databases. Thanks to our neighbor databases, they have developed a very good databases, and US or our own country imprat is a very good database now, where we can just type our botanical name, in fact the Ayurveda name of a herb, and then they will give you the n number of phytoconstituents. The big problem in that whatever been reported from any geographical area for that herb would be given in that database. Let's say I'm giving Ocimum centumum from India, may not have a phytoconstituent which is actually available in the same plant in Brazil, but it would be reported in that database and you will collect it from that database. Then there may be a high chance that you may be working on a phytoconstituent which actually is not present in your Khari Bavli or the near area Ocimum centumum. So this is a limitation for database use. So we are using another type of analysis where we usually do a TLC followed by mass spectrometry. Then we got some good somersaulting lines, and then we make a filter. And this again a very judicious filter. Which peak would work? We don't know whether it is a primary metabolite, whether it is a secondary metabolite. And a Dravyagun person could better tell us that yes, this although the peak is low, the the concentration is low, but it can potentially work. And depending on the previous publications also we filter such phytoconstituents, and then we do the ADME and our keys are ready. This part is okay. We do two ways. One is MS TLC MS, then we got such somersaulting peaks, and then we do the ADME. Here also we do the ADME, and then we just take the bioactives, or it is called the keys. How we trace out the protein? We just take uh, let's say someone is saying that I want to work on COVID-19. Recently we got published in monkeypox, so we know nowadays, we know that which protein is actually target protein in the body for any drug for preventing or curing the monkeypox or COVID-19. In case of COVID-19, we know that ACE inhibition was the prominence in those days. So we just took that ACE, and then we go into the PDB database, download the structure of that protein. Then a protein has a big structure, then actually workable area, we just we catch that subunit, and we just put that subunit into the software, and protein preparation is being done. Then the area where actually, let's say this is an whole lock, and key has to be put in that small area, then computer make that area more prone for that key, and it is called grid preparation around that key area. That grid is prepared, then we just introduce that ligand or a key, and this is called molecular docking. We got a particular dock score. Sometimes residents came to me and then says, sir, the doc score is 4, -4, -10, -11, -14. Is this good or not? So the genuine answer is, kindly compare it with your known drug. If your known drug, let's say in case of ACE, if SCQ is giving the score is -7 and your quercetin is giving -8 or -9 or -10, means your quercetin is giving good doc score than the SCQ or any other synthetic drug. So this is the pattern how we score. We score the likeliness of that phytoconstituent. So one way is docking. This is our picture where we can show you that this is an uh, a thrombin protein, and we have kept our durva in it. You can check it that key is moving, but protein is lock. Now in the rightmost corner, you can see the score. When the OH bonds are being changed, you can find the score is changing, and then the computer can tell you that it is the best score, or this is the best structure of your phytoconstituent which can possibly work and give a best score. I think this video was visible. Joshi sir, this video was visible. Hello, am I audible?
Yes, sir, you're audible, but screen is blank right now.
That video was visible?
No, previous video was visible, but now the screen is blank.
Okay, no problem, no problem. Okay, sir. So I'm just trying to tell to the listeners that here you can find the protein was fixed, but the key was moving. But in real time scenario, this does not happen. Protein is also moving in the body. So just after doing that, uh, this docking, you have to perform a dynamics. And in dynamics, I'm not going into the details of this physics or the computational approach, how we came into the dynamics. I'm just giving you a glimpse that how dynamics works in computer. And this is real time. This video will show you one thing. Here you can find the protein is also moving and the phytoconstituent is also moving. They are searching the best pocket, and once it got a best pocket, then it restructures to stabilize the that bond. So this is dynamics. Although dynamics need a GPU and high computational hardwares and softwares, but yes, few good labs are working now, and among them one is ours. Let me take an example. When you make a handshake with someone, or you just have a have a have a introduction with someone, a picture is being taken, and that picture of just having a hand around, and this is a single picture which gives you a docking. But dynamic shows you that how long your hand was shaking, in which direction that hand was, or those hands were moving. And another thing, how people were thinking when they are having a handshake, how their internal structure was behaving, their their protein axis was moving or not, whether they are moving from their places or not. This is dynamics. This is dynamics and above. So you can find in this picture that dynamics gives you that this tryptophan of 732 is being interacted for nearly zero point 0.2 nanoseconds, and this bond was basically a water bridge bond. Here you can find a hydrogen bonding. So in dynamics you can show that this type of bond was visible, and how long it was visible. You can see this is your key and this is the protein. You can see that OH is being making a bond with GLN, and this bond was for if we are doing the dynamics for 10 nanoseconds, 64% time this bond was visible. 62% time means 6.2 nanosecond time this bond was visible, and they are interacting with them. The best bond is this aspartate bond, which is actually built among between the OH and the amino acid, and majority of the time this was existing. So this type of picture actually been produced from the dynamics, and this is very very important nowadays. This is our own lab, this interaction bonds. Then coming on how your key was rotating, this is also one type of beautiful interpretation of dynamics. Coming on to the third type of technique, which is called network pharmacology. So if it is not overflowing, then I will continue. Otherwise, I'm not sure whether the people are interested to learn third technique. My time is permitting me to do. So,
Sir, you can
network
continue.
farm.
You can
Okay, sir.
continue,
Okay,
sir.
sir.
This is also
Thank you,
part.
sir.
Uh,
Okay. Okay. Let me give you a few glimpses and some hypothetical situations why we need a network pharmacology. Let's say one day Charak says, yes, these three things would be called Trikatu. These four things would be called Balachaturabhadra. These five things would be called Tran Panchmul. Is this the imagination or an observation? For sure it is observation. For sure it is a research product. Then only the Tran Panchmul Kush Kash Nal Darv Kandek Shugiti Tran Panchmula, जो भी whatever the shlok is, but these 5 things they have given. Let's say they says these 4 drugs, these 4 herbs are for nootropic activity. Actually, when such type of formulation are made, we have very less evidence that why only these 3. Why these 3 in Trifla? Why these 3 in Trikatu? Why these 4 in Bal Chaturbad? Why these 5 in uh, Panchtranmul or Dashmul or something? Is there any synergy among them, or they are inclining towards a particular type of disease or particular type of pattern or particular type of prevention? Network pharmacology can definitely benefit in it. I'm giving you a simple example. The story starts in 2019 when I joined Raipur, and one of my scholar asked me, "Sir, can we do a research on combining all 4 Midya Rasayans for a moderate type of MR, mental retardation?" Then we says, "Yes, why cannot we do this? Yes, we'll merge them 4 and then we'll check it." I was also very new in those days, and I had very less experience of PG guiding. Then when we represented it in front of the IEC people says, or IRB people says that, do show us a classical reference where all these 4 are being given collectively. We searched a lot and we did not find even a single reference where only these 4 are given collectively for nootropic activity or for middeta or for mental retardation. Then this is, if it is not in reference, then go for toxicity first. And then came the network formula. I just asked them whether if I can give you a computational result, should you accept it? Then this is okay. Then we did a protox and then we produced and they permitted us. Meanwhile, we thought, yes, if we are doing these all 4 or clubbing them all, is there a synergy among them? Then we started a network pharmacology, and till date we have at least 20 to 30 or 50 network pharmacology we have done till date. In network pharmacology, what we do, let's say we are taking Centella asiatica, Glycyrrhiza glabra, Tinospora cardifolia, and Convolvulus pluralis. Centella asiatica has these number of bioactives. This Tinospora has this, CP has this, and Glycyrrhiza has these number of phytoconstituents. We just collect them from the, maybe by HPTLC or LCMS or by database. Then what we do, the next step is, these bioactives are known to us, and thanks to the databases, how these bioactives works in the body is also known. How many proteins they touch or how many targets they touch is also known. So these green colors are their targets. Let's say this is an X bioactive, it works on EGFR. This is another bioactive which actually works on NOX4, and this is a pattern. So just going by, another next step is, these proteins are being actually involved in these number of diseases. So now we can see that having a formulation, they are herbs, single formulation, they are herbs and they are bioactives, then bioactives to targets, and then targets to disease. And club up is this Medya Rasayan, then these 4 herbs, their phytoconstituents, their common targets, and their diseases patterns. This is a particular presentation for a nootropic activity. If we club up these, now you can see they mean, if we are using Glycyrrhiza glabra and Tinospora cardifolia, there is a sync. When we did it for Balachaturabhadra, we were very, very happy that yes, majority of the targets are antidiarrheal. Majority of the targets are looking for dantodbhed vyadhis. It is not by chance, by choice, or just by dreaming that yes, Balachaturabhadra, these four, Karkat, Shringi, Aruna, and all these four are not been a single day dream of a of a acharya. There is a sync among these herbs, and this sync is actually can be predicted by network pharmacology. Let's say someday someone asks me that Karkat Shringi has been extinct, or let's say Shringi, or let's say Trifla mein Amalki is extinct. Which disease we should exclude from the shlok? I'm not saying that we can tell, but yes, we can predict that this shabd or this part of the shlok should be deleted now, because we now do not have Amalki in Trifla. Or one day we can also predict, yes, if we do not have Marich in Balachaturabhadra, or if we do not have Piper longum in an X formulation, we can replace it by using this herb, because the phytoconsonants are near and they are inclining towards the target which is actually been given in the shlok or in verse. So this is a remote and possible doable thing for computational or network pharmacology, for enriching our siddhant, our shastra, our samhita. Days are not very far when you would find a shlok derived from network pharmacology. We are working on it. Possibly, you just give us a Anubhut Yog, we'll give you a shlok. I'm not saying that this would be a best shlok, but yes, we can do it. Now, I'm giving you an example how this computation helped us. Government of India has given a task to us. Ministry of Ayush has given task to us. Yes, the pediatric vaccination is not available, and in third phase there is high chances that pediatric population may be involved for COVID-19. So that Ayush squad should be converted into syrup. And our department head, Professor Rajgopala sir and Dr. Arun sir, has dedicatedly developed that syrup, which is actually jaggery based syrup and more palatable to the pediatric age group. And when we developed that syrup, the question was, will it work? And can you give us evidence so that we can directly propose it for at least for prevention, not for the therapeutics? Then we did HRLCMS. Then same we used Lipinski, Weber, Ghosh, again Muges and kinetics and dynamics we did. And in case of prevention, there is a problem in it, because you don't have protein for prevention. We know protein for disease. For prevention it is very difficult. So we did two type of patterns. One is pulmonary protection and second is immuno enhancement. You can see these two figures. Well, one is pulmonary protection, second one is immuno enhancement. And we did a network construction, then docking dynamics, then gene ontology and cat pathway analysis. And then we said, out of this syrup, these four are possibly working for these two type of prevention: lutein, camphorol, hydroxycinnamic acid, and ginseng, and carbolic anhydrous too. If you do not have this syrup, please use these phytoconstants wherever it is available. Maybe if in X herb these four are available, use them. We did not say it, but at least we said that yes, these four are possible bioactives for such type of disease prevention. Then we also propose that these four are working through endocrine resistance pathway or estrogen signaling pathway. This with the help of gene ontology and KEG pathway, and our team, our lab did work very good, very. We did it in 20 days only, and then that we almost we distributed almost 1.5 lakh kit of Balaraksha kit, which has this syrup all around the India, starting from hills to the coastal areas, from northeast to west of Gujarat. We almost distributed 1.5 lakhs kits for that. We have encouraging data also. Maybe any other day we'll discuss that. But as of now, this single computational study has supported us to an extent that we could serve the society in a such a larger way. Because faculties are sitting here, uh, I can extend my presentation to the next level, which actually for the faculties. Uh, do we need in silico studies? Sometimes people says the biopiracy. Sometimes people says, why do we need it? It's very simply, it's very predictive in nature. What went wrong if we are creating an evidence? And why we are talking about biopiracy? It's already in existence. Things are already in existence. Those who really want to capture or catch or maybe pirate, they are, means I don't think that computation is helping it. They have their tools. Are we really ready for such coalescence? Are we really ready for such things that yes, informatics should be there, Ayurveda should be there? And if yes, then what do we need then? We need four things, which I actually discussed in initial first or second slide: that hardware, software, skilled human resources, and followed by institutions. Does available tools are sufficient? This is a very $1 million question. This, if the tool is, whatever tool we have, are they sufficient? Does available human resource are sufficient? What would be the roadmap then? So the simple reply, why do we need in silico studies? Yes, everyone is doing nowadays. Everyone is doing. There is a bargaining of globally bargaining for that in silico studies. They are cost effective. Yes, you can predict. Let's say someone is telling you that Trifla works in CA, breast cancer. Then keeping all phyto constant, taking them to the wet lab and searching it, it would take at least 40 scientists for 40 years. Using computation for 2 to 3 months or 4 months or rigorous 4, 5, 6 months, you can predict that these and 5, 7, 10, 15 phyto constant could possibly work in CA or whatever the your choice of disease. So you are actually preventing the time of the researcher, the money, and possibly the efforts. It could be the first predictive approach for reverse pharmacology in Ayush. Every time every Tom and Jerry ask us, do you have evidence for this drug? Do you have this and that? So computer is the easiest way to create an evidence now. Can reduce it can reduce wet lab load. Yes, a new interface for drug discovery. Believe me, whether we will do it or not, people are doing it, and one day this treasure of herbs would produce a number of natural, semi-synthetic, and synthetic moieties. Personalized prediction in medicine, this is also possible. I'll not discuss because it will take another 15 to 20 minutes. Why we ready for the coalescence? Dependent readiness. Just 4 to 5 years back when I was studying it, no one was working on it. Few people from Pune, few people from maybe IGIB or some people were working on it. Dependent readiness, yes, we used to request every informatic fellow that please do this doc or dynamics for us. With God grace and the director's grace, we developed such a lab that now we can do anything in a single click. Yes, our softwares or the available softwares, are they sufficient for large phytoconstants? This question is still persists. Maybe in next 2, 4, 5 years we can reply it, because if we are consumer and we have a number of consumers, the producers or the software developers will develop such type of softwares also. No academic training in the interface. Till 2021, there was no official academic training for Ayurveda and informatics. Then we developed a course of 7 days, and I'm happy to inform you all that we have trained at least 120 uh, Ayush researchers over this interface through three in silico trainings, which we did it in our lab. It is in 7 days workshop, and usually every year we do. This year it is nearly in the December. So those who are interested, they can check our website and they can fill. We have only 15 seats for outsiders, 15 for our faculty and students. We require softwares, yes. If buyer are more, the producer can produce any type of software. We need researchers over this interface and tailored training program or courses. We are developing it. We have developed SOPs also. We have developed a very beautiful uh, guideline that how a Ayush person should go in computation. This is published in Phyto Medicine Plus or something, which is also a very good journal. We this Sapphire guidelines, we Gupta and Jacha guidelines, we published with the help of Niper. Yes, much evolution is required. Tools are not sufficient. Traditional medicine centric databases are not there. Computational methods of modeling of Ayush is not available. As of now, not available. It needs a synchronization and systematic integration of data obtained from different disciplines of the science. So different disciplines are working. Ayush or Ayurveda has to work on it, so that a centric data should be available for computer to react or to sync or to simulate, or for the machine learning also. This actually a figure which we dreamed almost in almost 5 years back, when we said this central policy push for CCRS, Ayush and ICMR, then institution of integration should come up, then dedicated center for advanced learning, and in vitro in vivo support should be given to the Ayurveda plant phytochemical databases. Then the database would develop, then subject based in silico models. Nowadays, yes, herbo minerals we can do. Initially we were able to do the herbs only. Now minerals can also be in our lab, we can do that in silico models. Then the international collaborations is possible. Then Ayurveda centric in silico studies, concept centric. Someone uh, from the audience maybe in 2-3 years back, someone asked how you will incorporate Atma, Manha in computation. I just said that be a consumer. One day the software would come when you need not to, you would not be able to say these words. We have AI. So if we ask a genuine question from a software developer that yes, I want Atma in this, they will give you. Just the point is we have to precise our research question. Software is not far away. When AI, what AI do? It predicts you and behaves. I'm not saying that it is a mana or something, but yes, if you ask, they will develop. And herbo mineral in silico studies, and then only we can develop a better lead development. There's some glimpse about our lab. I have 5 to 7 minutes. This was inaugurated by our own the Professor Tanuja Nesri Madam and AIMS New Delhi Director Srinivasji, and our respected Secretary Sir Dr. Rajesh Kotecha, Vaidya Rajesh Kotecha ji. And then onwards we started working on, and we have a dedicated hardwares. We have multiple GPUs, CPUs. We have paid softwares of nearly 30, 40, 50 lakhs rupees. And we have database access also. This is our first batch. And you can see good faces from all around India which actually participated, and we did it in 2023 in February. And see the second batch, we have such diversities. 16 states represented our 30 30 student capacity batch. And you can see few of the very good faculty from your area also. Professor Patil and Professor Bhargav ji is there, and Professor BM Singh Saab, and Professor Sangeeta madam, our director madam also, uh, uh, Professor Manjusha Rajgopal madam, and multiple faculties participated in, even from Northeast. Few of the publications, this publication I discussed. Then this is a very beautiful conceptual paper which we published almost 3-4 years back, when we said yes, there is a possible in silico Ayurveda correlation. A confluence is possible for better development of traditional medicine. This also we published from Raipur and Ayur informatics lab. Then this is again a very good paper which actually I discussed for immunity enhancement, pulmonary protection of polyherbal Ayurveda. And then a very beautiful paper where we actually propose that if the patient is suffering from methicillin MRSA, which phytoconstant or a herb can be given along with antibiotics just to promote that antibiotic action, or just syncing them to prevent the patient from MRSA. This published in Chemical Biological Interaction, a very good journal, which is nearly more than 5 impact publication. Me and Dr. Mahapatra sir was the authors of that paper. This is overall computational biology work which we can think of. Yes, there is a software tool development, database development, software and tool application. System biology we always see or work on systems biology. Computer has a limitation, it works one to one, but in our case polypharmacology is required, so maybe one day we could able to do it. As of now we are doing network pharmacoinformatics, additive and substitutive informatics, ADME, molecular dog, dynamic QSR, homology, virtual screenings, pharmacophoring. We are doing this. uh, Thank you very much. I think this is from my side. Uh,
Thank you so much, Prashant sir. So,
I'm
partic-
very sorry if it is boring. I'm sorry because
no, not at all, sir. Not at all. It was very interesting, and there are a few questions in the chat box. So,
okay.
I'm
Let
going
me
to
see
read
some.
for you, sir.
Oh,
I'm
मेरा
going to
ये
read for
बंद हो.
you, sir.
Sir, please.
So, Dr. Tripti, she has asked, what is QSAR methods?
uh, Dr. Tripti, I would respect this question. uh, when which branch you belongs?
Probably Rasa Shastra.
Okay. So Dr. Tripti, I would like to reply in a very nutshell that when you say that CRP, C reactive protein, when you go for an infective cause and you just send a sample of a patient and this is C reactive is positive, okay, and sometimes you find a value in it. Yes, that C reactive is 15, 18, 20, 25, 30, 50, 4, 1. When you are quantifying the things, it is QSAR. When you are saying qualitatively, it is other techniques. This is the simplest way. We can take another lecture on QSAR. This is just an umbrella. Believe me, I am also very willing to tell you how QSAR has to be worked, but this is computation. Unless you do it on computer, you would not learn a lot. With due respect, I can extend for QSAR, but QSAR then it will take another 1 hour. So if someone permits me, I have a prepared PPT, but then you have to keep your desktop ready for it. or in fact, if QSir is of your interest, I will share my PPTs with you. Kindly share me your email ID, and
Sir,
then we will discuss.
sir, if you could share it, we have a LMS. We will share it in LMS.
Yes, sure, sure, sir, sure. It's already prepared, sir. I have nothing to do. मैं तो just share करना है, sir, मुझे. और किसी को प्रश्न होगा, then we'll discuss it. Next प्रश्न, sir. तृप्ति जी, अभी
Yes,
उतना
sir.
ही जवाब दे पाऊंगा मैं.
No
मतलब
problem, sir.
साधारण वे में क्वांटिफाई करना है, तो अभी अभी बस इतना ही कि साहब ये दवाई का असर होता है. हाँ, कितना होता है, कितना होने के लिए क्यूसर पे जाना होता है. भाई, बहुत ही लेमैन में मैंने आपको बात बताई. नेक्स्ट प्लीज.
Ji, another question is again by Dr. Tripti. Can we use predict compound toxicity software for Ayurveda herb by using their active phytoconstituent?
Yes, madam. For sure we can do. And what, with God grace, now they are free of cost available. Protox is free of cost available. You are welcome to our next coming workshop of 7 days. We do 3 hours hands on toxicity prediction. But the problem is that each and every phytoconstituent would represent uh, means in a isolation, that every phytoconstituent will be given separately. That club up formulation would not show you that yeah, this is toxic or non-toxic. Each phytoconstituent you have to put, and the result would be there in front of you. If at all any phytoconstituent so toxic that either it is coming under OECD or something, then this is difficult directly to use it in humans. But for sure we can predict the toxicity by taking help of softwares.
Thank you, sir.
Um,
Another question is by Dr. Rohit Johari. He has asked, in Abhav Pratinidhi Dravya, can a study be designed of targeted drug biomarkers to certain specific diseases? And he
Sir,
has
in
suggested some
Abhav Pratinidhi Dravya, okay. Can a study be designed of
biomarker.
targeted drug biomarkers to
Hmm.
certain specific disease like Babul and Bakul? Yes, 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 for sure. 100% कर ही रहे हैं, हमारी लैब कर रही है. मतलब हम लोग कर रहे हैं. तो डॉक्टर जोहरी जी, आप जहां से भी हों, we will share you our email ID of the lab. You just precise your research question, will help you in. अगर आप ऑल इंडिया से हो तो आ जाओ, आप तो कभी भी आ जाओ भाई.
Dr.
Yes, we
Par,
can do,
hmm, sir.
sir.
Dr. Parul Agrawal, she has asked how to get similes of different biomolecules.
So okay, that's great. Do you really want to read me who is Prashant Gupta? What you do, you just type a name Prashant Gupta in Google, and if you want to really precisely searching, then add Prashant Gupta Ayurveda. Same thing has to do in Smiley's search. You want to search Smileys for curcumin, type type curcumin in Google and just add Smileys. You would see, if you can see my slide, just a second, sir.
Yes, sir.
Just
Another
एक
question. Okay.
एक मिनट
Are
sir.
you trying to show it on screen?
Yes, let me show
Yes.
it, sir.
Yes, sure, sir.
Let's say curcumin Smileys. Okay. Okay, so see this. PubCam is available for you, and you just see it. So here is 2.1.4 smileys. You just have to copy it here. Okay, I have copied it. Let me show you another very beautiful thing. Swiss ADME, which is also free of cost available. Dr. Tripti, you can see it, I think. Dr. Sorry, Sam Agrawal. Okay. I had copied the smileys from curcumin, pubchem. Swiss ADME would take some time because this is इस पे लोड बहुत रहता है. Okay, you just have to put this smiley control V and simply run. Or if you have n number of, let's say you have 50 phytoconstant, copy them and put in single way and just run. You can see this picture. GI absorption high. If you give orally the curcumin, it is absorbable. Blood-brain barrier, no blood-brain barrier crossing. Moderately soluble, yes. And log P value is -6. Yes, if you apply it on skin, it could absorb. The Lipinski, Ghosh, Weber, Egan, Muges all qualifies. Synthetic accessibility means if you try to structure this in the lab, it is possible that it could be synthetically constructed. If the value is high, it's very less likely that every phytoconstituent you cannot make in lab. And one day if this curcumin is working in, let's say in pain killing, how much curcumin you need, then you have to construct it in the lab. And this score synthetic accessibility means that whether this phytoconstituent can be developed in lab or not. Nature can produce any twitch, but we cannot. So this synthetic accessibility shows you that yes, it can be reproduced or it can be developed in the lab. If the score is 10, it is very difficult to develop in the or make it more stable or stable in the in that form. So I think it was visible, है ना? So smileys, getting smileys, let's say any other name you want to take, I can do it here. Let's say vasacin or anything, let's say quercetin, pubchem. You just see it if it is visible. Yeah, this is smiley. Just copy it from here. It is as simple as copy and paste. You just put it here and just run. Everything would computer would do everything. So this is again another type where you can see okay, I think I have not copied it. Same smileys है क्या? I think you now know that how it works. I think your query is clear then.
Yes, sir. So uh, the next
When is
question is, yes, sir.
yes.
Please, sir.
When is the next training intended to be? Dr. Jyotsna Nair, madam. Okay, I
Yes, sir.
don't know whether you are faculty or a professor or a student. It is
She has
open
asked
to.
me, is it open for the current PG scholars? That is what she has asked.
We are really very sorry that as of now we are not opening it for the PGs because this is very dedicated 7 day cumbersome 12 hours training every day. So it is open for faculty and PhDs. PG scholars, we can construct it or structure it if we require or demand arises. What is the selection process for the candidates? Okay, selection process we cannot declare it here. This is our internal policy.
Okay.
Is Agray Dravya prediction reasoning logical possible with these techniques? Yes, Anish ji, yes, 100% it is possible, sir. That's why we are developing. Please share the Google Classroom. How to join Google Classroom?
So
Okay.
that is related to us, sir.
That is related to us, sir.
Okay, sir,
We
sir,
will
sir.
share the link.
Okay. So thank you, sir. I am very much interested for this workshop. This is my field of interest. Will join soon. Most welcome, madam. Are the softwares of network pharmacology open for use like Swiss ADM and Protox? Forum Shaji, yes. Yes, 100% network pharmacology is free of cost available. रोहित जी दयानंद जालंधर से हैं अच्छा अरे वेरी गुड पारुल जी थैंक यू is Ayur informatics belongs only to database on drug development in other ways does it include storage of ebooks e-samitas अरे साहब उसके लिए बहुत बड़ा ग्रिड है हमारे पास e-samitas bibliometrics we have big things for it yes informatics has multiple dimension HIMIS में भी लग सकता है ये बहुत it informatics can be utilized in n number of ways the the single thread which I had discussed today is computationally aided drug designing, sir. Can we identify vipak of food grains, the fruits, the vegetables which are not well mentioned in the granthas? I would request Madhumati Navkar ji, please come forward and help us or do a independent research for it. If you need help for us from us, we are here to help. But as of now, uh, we developed only one Rasgun Veera Vipak network pharmacology till date, which we produce in front of the some some officials. Possibly, possibly we can do it, but then we need dedicated people like you or dedicated faculties for it. Many times major phytoconstants from plant are excluded after ADME analysis. Yes, which having main action what should we do? Is it necessary to subject plant based phytoconstituent to ADME? Oh Tapasvi ji, it's very important. I think acha, okay. Dr. Jitendra ji is from All India. He is a scholar and is a very good bio this bioinformatician also. This question is very valid. Phytoconstants should not be excluded because phytoconstants are large and these ADME exclusion are based on the molecular weight and number of carbon or number of sulfurs, nitrogens. So this is a very pragmatic question. We are trying to search it. Maybe other type of ADME is required for phytochemistry or phytochemicals. So this question, yes, we need a dedicated software. As and when it is available, we'll do it. Can Swiss ADME work for compound drugs? Compound drugs? Mayank Kumar ji, compound drugs का मतलब? ओके छोड़ देते हैं. कंपाउंड ड्रग्स मतलब मुझे लगता है फॉर्मुलेशन ड्रग्स शायद कर सकते हैं. Dr. Tripti, sir, is there any range for skin permeability in Swiss ADME? Yes, there is a range. One says yes, skin permeability is there, and some says no. Access whether a drug can be used topically or not. Yes, for sure we can do it. Excellent lecture. Thank you, Bhaiya. Pradyanya Gathe. Dr. Shivani ji. Okay. Madam, namaste. Wonderful informative talk. Thank you, madam. Jyotsna Nair ji. Is there any possibility of collaboration for independent researchers with AI Lab? Yes, most welcome. Kindly write to us. We'll take a due permissions and then we'll welcome you at our lab. Okay, recording. Are these smileys notations linked with the ras dravya like dhatu, padhatu, ratna? No. See, ras dravyas are basically, if you talk about bhasmas, bhasm is not just metal or mineral. There are phytoconstants because you just give, when you give a triphala in a bhavna, it is in the final product. Then you have to do XRDs, you have to do ICP-MS, you have to do real-time X-ray crystallography, you have to sometimes NMR also. So it is not easy, and this is our problem. So we have to take and reply. We have to create and reply. So in such cases, I request that when multiple researchers would come on the surface, such problems will be solved. Otherwise, as of now, dhatu, upadhatu, ratnas, yes, it can work. We can keep this particular type of metal into the dock or dynamics, but ratnas I have not attempted. I'll ask to my Russia's fellows how we can incorporate or we can introduce these things uh, into the computation. How to connect you, sir? Can you share the email ID? Yes, for sure, madam. Let me write how I can write. May I write the email, sir? Joshi, sir?
Yes, sir.
Sure, sir, sure.
You send message to us, sir. We will put
No,
in
you
the.
can directly put it in chat box. No problem, sir.
Sir. Okay, sir. Let me know if anything from my side then.
No, thank you, sir. I request Supriya madam, are you here? Would you like to add something or?
Supriya madam, namaskar. Shivani madam, namaskar. I think Supriya madam aa gayi.
Sir, Joshi sir, uh,
Yes, ma'am.
I miss, there is one more lecture going to happen on bioinformatics. Uh,
Yes.
so we will have some discussion after that.
Okay, sure, sure.
Okay. When is that lecture? Please let me know also so that I would also attend it.
Yes, sir. Yes, sir. We will share you the entire list.
Sure.
Uh,
Thank you, sir.
Now I request Dr. Vikrant to just summarize and thank sir. So thank you very much, sir, for joining. Uh, इसी बहाने यह फैमिली दिल्ली से भी ज्वाइन हो गए बहुत सारे एंड आई जस्ट वांट टू इन्फॉर्म कि ये सेशन चलता रहेगा. इट विल बी कंटिन्यूड अप टू 17. तो 17 अक्टूबर तक है एंड इट विल कवर वेरी इंटरेस्टिंग टॉपिक्स. जेनेटिक्स में भी देर विल बी 4 to 5 सेशंस ऑन जेनेटिक्स, कॉन्जिक्यूटिव सेशंस. सो ये एक अपडेट है. द डिटेल्स विल बी इन्फॉर्म वेरी सून. And those who have not joined Google Classroom, they can join. The link is posted in the chat box. Thank you, sir. Thank you again.
Sir, may I leave now, sir? Okay, sorry. Please. Please, sir.
Dr.
Continue.
Prashant Gupta, sir.
Thank you.
Thank you for your thorough lecture. You have gone through the basics, hands-on training, molecular docking, that is lock and key method, and the picture, the demonstration was very good. The molecular dynamics, and you have differentiated the within what is meant by molecular docking and what is meant by molecular dynamics. Then network pharmacology, the need of Ayur informatics. Sir has opened a new door for all of us in our research work. Thank you so much, sir, once again, and thank you all for the of the all the participants for your attentive listening, and thank you so much. Thank you, sir.
Thank you very much, sir. Thank you.
Thank
You
you,
very much, Joshi
participants,
sir.
for
Thank you,
joining.
Sujata
We,
madam, and thank you everyone, sir. Thank you.
yes, sir. All the participants, thank you very much for joining. We can understand this. These are the late hours. Some of you are in OPDs, clinics. Still, you are finding time and attending these sessions. Probably do encourage all your students to join. Uh, there is no issue of capacity. We can extend the capacity up to 1,000. No issues. So thank you again, and we are signing out.